Escherichia coli, case study by Shayna and Adrianna. Shigatoxin producing E. coli, 0157, specific strand name. Shigatoxin producing E. coli, 0157 is a specific strand of bacterium, Escherichia coli, that we have spent the past few weeks examining in class and lab. Shigatoxin producing E. coli, or shivatoxins, are a group of toxins associated with producing severe illness in humans. 0157 refers to the specific antigen, which is part of the cell wall. Antigens are used to help classify the specific strand. 0157 is the antigen found on the surface of the strand.
E. coli stains gram-negative in a gram stain, just like every other strand of Escherichia coli. Shigatoxin-producing E. coli 0157 is composed of gram-negative bacilli. This means that they have a thin peptidoglycan cell wall and will stain pink on a gram stain. Peptidoglycan is a polymer compound composed of peptides and polysaccharide chains, and the function of the peptidoglycan cell wall is to support and protect the cell as well as anchor other components of the cell to its membrane. The cell membrane contains lipopolysaccharides and also helps to contribute to protecting the cell.
Viewing the microbe. There's 10 steps into viewing the microbe. We have prepare the microscope, clean the lenses, select the appropriate objective lens, prepare the microbe slide, place the slide on the microscope stage, focus on the microbe, adjust the magnification, adjust the illumination, observe and record, and clean up. But in steps, we have prepare the microscope. We prepare by placing the microscope on a clean, flat surface. We plug it in or ensure that it has a power source. Turn on the microscope's light source if it has one. Next, we clean the lenses. We use lens paper or a microfiber cloth to gently clean the microscope's objective lenses, the one closest to the specimen, and the eyepiece lenses. Clean them if they are dirty or smudged. Then we have select appropriate objective lenses. Start with the lowest magnification objective lens, usually 4x or 10x, for initial focusing. You can increase the magnification later for more detail. Next, we have prepare the microbe slide. If you have a prepared slide, skip the step. If not, prepare a slide by placing a small drop of liquid containing the microbe. Example would be water, culture medium, or a stain solution, onto a clean microscope slide. Place a cover slip gently on top of the liquid drop. Make sure there are no air bubbles. Then we place the slide on a microscope stage. Carefully position the prepared slide on the stage of the microscope. Secure it in place with the stage clips. Then we focus on the microbe. Look through the eyepiece and use the coarse adjustment knob to move the objective lens closer to the slide. Bring the microbe into your field of view. Once the microbe is roughly in focus, use the fine adjustment knob to sharpen the image. Then we adjust the magnification. If you need more detail, switch to a higher magnification objective lens. Example would be 40x or 100x. Be careful not to touch the slide with the lens, or the slide may become damaged. Adjust the illumination. Adjust the light intensity using the microscope's control to achieve the desired brightness. Too much light can wash out the image, while too little light can make it hard to see. Now we observe and record. Observe the microbe carefully. You can make observations, take notes, or capture an image using a camera attachment if available. When you're done, switch off the microscope and carefully remove the slide. Finally, we clean up. We clean the lens with lens paper or a microfiber cloth. Turn off the microscope and unplug it if necessary. Store the microscope in a safe and dry place, and then clean up your area.
Specifically to explain Anna's symptoms, we know that Anna didn't eat any food that was deemed to be contaminated, and it appears that her symptoms such as diarrhea would be caused by the shigatoxin. Since STEC 0157 infects the intestines, I can predict that her diarrhea is slimy due to excess mucus production. The intestinal cavity is lined with mucosal membranes that, when they are irritated or otherwise disrupted, will produce an excess of mucus in order to neutralize the irritation. This would cause the extra mucus to come out with her feces and therefore make it appear slimy.
Steps to treatment. No antibiotics, no antidiarrheal, monitoring IV hydration as needed. As the toxin is discharged when the cells rupture, administering antibiotics can exacerbate the assault by inducing the additional release of toxin. Antidiarrheal medications can potentially slow down the elimination of bacteria from the digestive tract. And that is our E. coli case study.